That might be a good idea. Maybe. Hold on a sec. I'm not sure. Because it might give you a warning, like if you leave it, you can't ever enter again. It might say that. So, all right, guys, we're going to get started here. Happy Friday to everybody. Good afternoon. Good afternoon. Okay, so a lot of you asked me about the clicker situation. One thing to note, if you didn't get an email from me on Wednesday, then you're good to go. Okay. Was it Wednesday that I sent it out for those of you that did get the email? Yeah, I think it was like after class Wednesday. I looked at everybody who wasn't registered and I sent you an email. Okay. So those people were still trying to figure it out. But if you didn't get that email, then you're fine. I just wanted to walk you through the process here on my phone. So you can see, you can do it on your phone right now too, and just make sure. So if you go to the iClicker, if you just literally just go to iClicker here. Just type that in. That's not how you spell iClicker. Yeah, so iClicker. Okay, then you're going to go to the menu up there in the top, and you can click on sign in. Okay, so if you're one of those people that got an email from me, just do this with me right now. Okay, you may have already done this, but we're going to give it another try. You got to choose students to sign in as. So I have a student account, so I can figure this out. Got my password saved in there. Okay, and I haven't added the course yet because I want to show you how to do that part. So you might have to, if you haven't done this part yet, then the first thing you might have to do before you see courses is choose your institution. Okay, so you'll choose Point Loma Nazarene University, and then you'll click on add a course. There we go, there's the institution. I'm going to click on Point Loma Nazarene University. Okay, and then we can just start typing in Bio 1030. There we go. So Bio 1030 2021, that's our course. iClicker remotes only. Okay, if this process doesn't look familiar and you think you registered it, then please do it with me. Okay, so I'm going to click there. I'm going to add this course. Okay, so remotes only. And then right there, you can either just click got it, or you can say, I already have your remote, register it now. I can click right there, register remote. And then you're just taking that ID number from the back and putting it in there. Now there's some of the letters are a little bit tricky. So make sure if it didn't work for some reason, make sure like your O's and your zeros look right. Okay, the zeros are definitely like a little bit taller and skinnier than the O's. Okay. Yeah, so just double check you got the right one in there, and then you're just going to click save. And then if you haven't set up your profile yet, you'll also set that up, and you can put your Point Loma ID number in there, that 6-digit ID number, not here, but in another spot where it asks for student ID. Okay. All right. So any questions on that? And who was just telling me that there was like an issue that they were trying to deal with, and they were going to try it that way? Who was that before class? No, that wasn't very descriptive. Somebody was just telling me they had tried it. There was some issue with the registration, and they did it a different way than it worked. You guys don't remember who was just talking to me? I don't remember who was talking to me. Yeah, well, no, I don't think it was you, Danica. Yeah, okay, all right, I don't know. But the one, yeah, so that's the way you do it though. So hopefully that works. And yeah, any other questions about that? So hopefully everybody will get in there just fine now. I will try to let you know for sure. And even if, yeah, it's not the end of the world. Like if there's some technical thing going on, I can always see it by your clicker ID number too and find you. It's just obviously a lot harder to do it that way. So while we're getting it figured out, it's not we're going to be okay if it's not your fault. Okay. So just try to get it registered though. Okay. All right. Okay. So before we hop into this stuff, next week is going to be our first, or on Monday is going to be our first TBL day. Okay, so again, what that means is you're going to study that video that's posted, okay, for Monday's class. Go to that week 3 overview page at the top where there's a little green section with the stuff for each day. Scroll over to the right if you're on your phone trying to do it so you can see it. And you'll see a little YouTube link, click there. You'll watch that video, fill in the notes just like you normally would, study that stuff well, and then come ready for the quiz on Monday. Okay, so you're gonna take a quiz by yourself. You'll send in the answers on your clicker, but you're gonna take it on a piece of paper like you would a normal quiz, and then you're just gonna plug your answers in on the clicker. I'll show you how to do that part when we get there on Monday, okay? And then again, you'll turn with your team, take that same quiz again, and then we'll do some activities. Okay, there's a set of notes after the one that says TBL day. Okay, it says TBL day activities. You don't have to study that before class. Okay, so just focus on that first one that the video is over, and then we'll get into the other ones during class after the quizzes. Okay, yeah. So anything else? Any other questions you guys have about next week? Go ahead. You're talking about the quiz on Monday? Yeah, the quiz on Monday is just over the stuff for Monday. Yeah, yeah. And then you got lecture quiz 2 tonight. Okay, so it's over those 2 sections of notes that we've already finished. And then, yeah, everybody hopefully had some fun in lab yesterday. We had Professor McGaw and I had fun hanging out with you. I know it's kind of information overload for sure. It's a lot of information taken in, a lot of new stuff. Were you able to show them how to get to the practice anatomy lab yesterday? Did you show them that? Okay, that's fine. Let me do that real quick. Yeah, no problem. Yeah, yeah. Let me show you guys that real quick because it's a helpful tool. Because the only time you actually get to be with the bones is those few times that you're actually in lab. Okay, so in your Master in A&P, I'm going to pull this up for you. There's a nice tool here that helps you. It just has lots of nice images of the. Oh, that's not going to work. Huh? There we go. Ah, gotta click on it. There we go. Okay, so you guys don't have that part of it, but when you get to the course, after you click in through Canvas, you'll get to something like this. And have you guys seen something like this, like a study area? It doesn't look exactly like this from the student view, but you're looking for study area somewhere. Okay, you're going to click on study area. All right, launch the study area. You got to click lots of little things here to get to where we're going. Okay, and then there's all kinds of things. There's all kinds of animations and videos, interactive physiology, all kinds of things. But the main one we'll want for the lab study is right here, Practice Anatomy Lab, PAL. Click on that. Got to click on this one here now too. Okay, and then under the human stuff up here, we have some pictures of the muscular system. So when we get to the muscles, this will be helpful. We have pictures of cat stuff too, which will be really helpful for you to study those cat muscles when we get to that part. But for now, we can go to these 2 skeleton ones. So appendicular skeleton. Okay, we can click a region. Let's do the upper limb because that's what your quiz is on next week. Okay, so remember your quiz is on just the upper part next week. Okay, so we got the bone there, humerus, right? And we can mouse over everything to kind of quiz yourself. Okay, it's a really nice way to quiz yourself, really nice images. All right, capitulum, trochlea, all those things. Okay, so it's got every possible bone, every possible structure there, more than you actually need to know. Okay, and the key thing for those quizzes is what's in those tables is what you need to know. Okay, there's lots of other structures and bones in the book and in this, you don't need to know that. You just need to know what's in those tables. Okay, and the format of the quiz next week is a PowerPoint with images somewhat like this. And the questions could either be name what bone this is, or it could be name what side this is, right or left. If it's a bone, you have to know that for. Or it could be pointing to something specifically, and it would say name the structure. Okay, and then the last type of question possible could be pointing to a structure on a bone, and it could say what articulates with this bone or with this structure. Okay, that's the one that's extra credit, sort of, not really, but you don't have to know those for the practical. But I test you on that for the quizzes. because I think it's just really, really helps you when we get to the joints that you understand where those two bones touch each other and where they come together. Okay, so those are the types of questions we'll have on the quiz next week. So you'll just have a blank piece of paper with numbers. You'll see the image up there. You'll write it in. You'll get a good amount of time the first time through, and then the slides will come through again, and you'll get a second time to look at all of them and double check your answers or yeah. Okay, so that's what the quiz will look like on Thursday. Okay, so big week next week, right? We got the TBL day on Monday, which is that IRAT quiz. We call it Individual Rapid Assessment Test. And then the TRAT, that's the team one. Then we go to the lab quiz, and then Friday is exam one. Okay, so lots going on, but it's good to have that perspective. Be thinking about all the stuff you got to do and studying, and yeah. So remember, on these Mondays that we have a TBL day, we don't have a master in AMP. Okay, so no master in AMP next week, but you do have that TBL day. Okay. Any other questions about what's coming up? Go ahead. So the lab quiz, yeah, the first lab quiz is on the upper appendicular skeleton. Okay, so I think that's called exercise 3 in the handout. I think there's exercise 3 is the upper one, then exercise 4 is the lower one, but it's just that first part. You worked on both the upper and lower yesterday, but we're just getting quizzed on the upper part this time. And then we're going to do the chemistry lab. We're not doing the bones next week. We're just going to do the quiz on the bones. Okay. And then when you come back the next week to lab, then you'll have the lower extremity quiz. Okay. And then we'll be back with the bones that week. Didn't work. Okay. Other questions while I'm getting this connected here? No. Okay. All right. Okay, so yeah, oh yeah, and one other thing for lab next week, and I'll remind you again when on Monday and Wednesday, but you're going to need to bring your laptop to lab next week. There's a couple activities you're going to do in that practice anatomy lab thing we just looked at. And so I'll give you another reminder, but at least one, you or your partner is going to need your laptop for lab next week. Okay, ready to go? Okay. All right, let's do this. So today we're talking about cell membranes, vesicles, and then all these little things inside of cells called organelles. Okay, so let's get into it. So we talked about phospholipids a little bit last time, right? And so phospholipids are the special type of lipid that make up our cell membrane. Okay, and this little GIF up here is nice because it kind of gives you the idea that the cell membranes are kind of wiggly like this, and they are. And so what it's known as far as the model of what cell membranes are, it's called this fluid mosaic model. And the fluid part is kind of called that because it has this kind of fluidity to it. It moves around. Things can squeeze through those little holes sometimes if they're small enough when they're kind of moving around like that. Okay, and then there's the mosaic part to it, which this is a mosaic right here, right? So it's a tile type artwork that has a bunch of different colors, a bunch of different pictures, all kind of put together. And that's kind of what all these different things that are plugged into the membrane represent, is all those different mosaic tiles you would have in that. So the fluid mosaic model. Okay, so the components of the membrane are free to kind of wiggle around, move around, but they can't like change positions, jump, but yes, we have these kind of wavy movements. Okay, so let's dive into these phospholipids a little bit more. We talked about them last time, but let's dive into them. So take a second, and you looked at this a little bit in your Master in AMP homework. We talked about it a little bit last time. So take a second with the people around you and see how many of these questions you can answer here. Okay, and I'm going to open up the clickers for, let's see, let's do it for that second part there. Answer this question on your clickers. Okay, go ahead. So turn to the people around you. Try to fill in as much of that with your team as you can.
Right? Because non-polar is always because polar molecules.
They like wiggle around, but they won't necessarily like go from like here to here or something. No, they like change positions.
Okay,
Yeah.
today in advance show,
Uh-huh.
we talked about the Mosaic
Okay.
model and about how these move so fast around and actually change all their positions.
Okay. Yeah.
I don't know.
Yeah. I mean, I think it's kind of just a different idea than what we're talking about.
Okay.
Yeah. So they definitely can be switched out. Is that kind of what we're talking about?
Well,
Like, so
saying
part of
you were
it
saying,
is.
like, that these literally move, like, I think you put it in terms of like 680 million miles per hour,
Okay.
but like that they
Yeah.
actually like
Okay.
move around.
Interesting. Yeah. 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 I think the main thing is the idea that like the proteins don't necessarily like
Yeah.
change positions super rapidly, but definitely the lipids are constantly always kind of moving. But I think the idea would be like this one isn't kind of like
No,
flipping
yeah.
places with this one. Is kind of what I was trying to get at. But yeah. Yeah. No, it's a good question. All right. So go ahead and vote here if you haven't. The one that I got the star by there, yeah. So you're answering the one with the star by it there, just which part is hydrophobic? Okay. All right, it looks like it might be the first day we have everybody in class. Almost have all the clickers, it looks like 68, 69. That might be it, yeah, okay. All right, let's see what you guys said here. Yeah, so yeah, about 60, or 97% of you are saying B here, okay, is the hydrophobic part, okay? So the tail, it would be the hydrophobic part for sure, okay? And then which part is polar, A or B? A, okay, good job, okay? So the polar head, the non-polar tail, okay? So this is a great example of structure dictating function, okay? So the head is polar, like I'm just saying, the tail is non-polar, okay? The head is hydrophilic, meaning that it's okay with interacting with water, whereas the tail is gonna push away from water. Okay, and this is a special type of a molecule called amphipathic. Okay, and what amphipathic means is a molecule that contains both a polar region and a non-polar region. Okay, so it's like an amphibian, and that's where that word comes from. Okay, if you're done writing, you can start thinking about that question on the next slide. I'm going to leave this up here for a second. And I'll open that up again on the clickers for you to answer that one. And talk to the people on your team about that clicker question as well. Okay.
And then like I'm being like, okay, focus on it. And then it-
Is that with Dr. Durrell, that class from it? Yeah, yeah, I'll have to ask about that. Sounds interesting. Never how fast it moved. I've never heard that
But he was
stat
talking
before.
about it, it's just saying that they're like constantly moving, they just don't really like flip flop.
Yeah, flip
But,
flop. Yeah,
yeah.
yeah. That's super cool. Yeah. All right, so send this in. Okay, we got 58 of you so far, 61. A couple more people, give you about 5 more seconds. All right, pretty clear. Pretty clear what the answer is, I think. Every time I do that, it kicks my camera off, and sometimes I try to watch the videos. All right, so go ahead and send something in if you haven't yet. Don't say whatever, figure it out. That's the whole point. Keep talking about it. Okay. All right, send something in if you haven't yet. Give you about 5 more seconds. All right, about a couple more of you send it in. Okay, so about 2/3 of you are saying, okay, about 7. Now everybody's switching over to C. I see ya, no pun intended. Yeah, so everybody's saying C. So tight junctions, yeah, we see something trying to get through there and it's not, right? That's what a tight junction would do. Desmosomes, we see these big flat things on the inside of both cell with some other proteins kind of connecting, right? And then gap junctions, we see things passing right through. So good, so tight desmosome gap, perfect. Okay, any questions on any of those? All right. Okay, start the video again. Cool, okay, all right. Okay, let's go ahead and answer this one. This would be like somebody takes your arm and twists it like that, right? What the bullies did to you in school, hopefully not, but. No questions allowed. Yeah, sorry, I'm just kidding.
Okay, can you explain? So when you said rivets, I don't really actually know anything about a rivet in real life.
Yeah, so it's, yeah, there's kind of like a flat metal thing there. You have that exact same thing on the other side of the metal there too. And then on the inside, just like a little, like almost like a nail that connects the two.
So it's like,
So it just pulls them together. It's wider on the outsides and yeah, like that. And so nothing can get through this way, right? And it squeezes together and holds those two spots together.
so that's this?
That's the desmosome, yeah. Yeah, it's kind of flat and wide on the inside of the cell.
Is this the flat and white part right
Yeah,
here?
exactly. Yep. Yeah. All right, go ahead and vote if you haven't. Only got 50 of you. Couple more. All right, 62. Give you 5 seconds. Need about 6 more people to send in their answer. Okay. All right, so most people honed in on C there. So they're held together by desmosomes. Okay, and that's exactly true for your skin cells. They're held together by desmosomes, keeps them really tightly bound so they don't get pulled apart. Okay. All right, so let's we're going to switch gears a little bit and talk about one of the really important functions of our cell membrane, which is to form vesicles. Okay, and so when it forms or when we want to do things like kick things out of our cells or get things into our cells, then the cell membrane can actually just function as a little transport system for doing that. Okay, and so we'll look at a couple examples of that. But when we're talking about exocytosis, that just means exiting the cell or getting out of the cell, right? So this is releasing substances out of the cell. Okay, so this would be things that we want to get rid of, like waste, mucus is important, but then also things that we want to put into the blood or into a space between a nerve and a target tissue, something like that. So neurotransmitters, hormones, all of these things are usually this whole signaling pathway that causes these things to be released. Okay, so that's exocytosis. But then endocytosis is the opposite when we're bringing something into the cell. Okay, and so there's 3 primary types. First type is phagocytosis. This is when the cell membrane literally kind of wraps around something large and then just pulls it in. Okay, macrophages are the most famous phagocytic cell for sure. Okay, and then we can do, cells can also do pinocytosis, which is just kind of taking a little gulp of the extracellular fluid. So kind of picture this little mouth forming on the outside of the cell, kind of just closes in on something and then brings it in. So it's not necessarily closing in on a single large thing. It's just taking a gulp of the fluid and whatever is in there comes into the cell. Okay. And then lastly, receptor mediated endocytosis. And this is when some type of chemical signal binds to the outside of the cell on a receptor, and that stimulates the cell membrane to then form a vesicle and bring it into the cell. Okay. So insulin works this way. Other certain viruses work this way. Okay. Clicker question again. Go for it. Which one's which? Talk to the person next to you.
Because wait, this one looks like it's peak hydrated. And then I think the last one's negative. And I can't even really tell what's
All right, so go ahead and vote on your clickers. Send in your answer. Okay, about 10 more of you need to send it in. Send it in on your clickers. All right. Anybody else? Got about 3 more people. Okay, let's see what you guys said. Yes, so 90% of you are saying D. Okay, so D would be pinocytosis first. Okay, receptor mediated. So the little red things are the receptors, right? And then those little circular things are the chemical signal of some sort. Okay, and then phagocytosis here with that larger thing being engulfed. And the cool thing about this is, as you can see, when the cell brings something in, it then keeps it in this lipid membrane, right? So now it kind of has its own phospholipid bilayer, and this helps in multiple ways. One way it helps is that in case that bacteria could potentially do damage once it got in the cell, it's in a little jail cell inside the cell now, right? It's stuck inside there, right? So it can't go and start messing with things. Also, other organelles can come up next to it, pump in some enzymes, pump in some chemicals, and destroy that thing before it ever has a chance to make contact with anything inside the cell, right? So it's a very efficient way to bring things into the cell. Okay. Yeah, let me show, I'm going to show a quick video here. This is just an example of phagocytosis. It's a fun one. You get to watch a neutrophil, which is the most prominent. There it goes. Yeah, one of our leukocytes, one of our white blood cells. It's chasing around that bacteria. You see it? It's going to catch it. Watch it. Keep watching. It's going to get it. There it is. There it is. Boom. It's gone. Yeah. So there's a neutrophil. Neutrophils are most abundant white blood cell, the most likely cell to make first contact with an invader. So yeah, just an example of phagocytosis there. Okay. Yeah, one quick thing, you guys, as I look around, most of you are doing great with this, but I will remind you that your masks need to cover your nose and your mouth, okay? Because if we got masks with our noses out, not really doing us any good, okay? Because then your air is coming out into the rest of the class. So I'll just remind us every once in a while to make sure. I'm constantly having to pull my nub as I'm talking, it's pulling down. So it happens to everybody. I know it's annoying, but we got to do our best to try to keep that up. Okay, thanks, guys. Okay, so let's switch gears and talk a little bit about the different organelles inside of cells. Okay, And so we'll start with the nucleus here. Nucleus literally means a little organ. Okay. Or sorry, the organelle means not nucleus. Organelle means little organ. Okay. So we'll put that here. Okay. As we think about the human body full of all its organs that have different functions, it's kind of like each cell is like that too, right? Each cell is this little body that has a bunch of little organs inside of it that have different functions. And that's kind of the idea of an organelle. Okay, so obviously the nucleus is where all of our hereditary material is. And what do we call that material again? DNA, of course. Okay, this is where we're going to be encoding for proteins. Okay, so we'll just kind of go through a quick overview of each of these organelles and their functions. And yeah, and then we'll do a little bit of a recap of the different ones we talked about. Okay, everybody knows mitochondria as the powerhouse of the cell, right? All kinds of funny videos on that, right? But yeah, produces ATP for us. We'll talk about that specific process later when we get to the muscle metabolism section. One unique thing about it is it has two layers of membrane. Okay, not just a phospholipid bilayer, but it has two of those. Okay, and so it has the outer membrane and the inner membrane, and there's tons of folds in that membrane on the inside. Okay, what's the answer for why those folds are so important? Go ahead. Okay. A lot more what? Okay, yeah, so it increases surface area, so we can do more with that surface area. Namely, we can plug a bunch of enzymes into that membrane, and we got more space to do that. The more folds we have, and those enzymes are going to be what drives things like the Krebs cycle and oxidative phosphorylation and all of those things. Okay, so the more membrane space we have, the more of that we can do. Okay, and those folds are called cristae. All right, yeah, so to increase surface area to drive things like the electron transport chain. Okay, so I'll let you keep writing there, give you a sec. All right, so next one, ribosomes, okay? Ribosomes are technically not an organelle. They're technically kind of more of this little molecular machine, but they're made up of both some protein and some RNA, okay? They're these little protein factories pretty much, and they're gonna help us to do one very important step in the protein synthesis process, which is called translation. Okay, we're taking that messenger RNA that was made in the nucleus, and then we're going to convert that into a string of amino acids, and that's what translation is. Okay, so translating messenger RNA into proteins. Okay, some of these are bound right to this part of the endoplasmic reticulum, and that's where it gets its name as the rough ER, because it looks rougher because it has all these little ribosomes bound to it. Okay, and then some are just floating around freely in different parts of the cell, so proteins can be made in different places. Okay. All right, so it's ribosomes. Okay, and then we have this whole kind of series of structures that all works together called the endomembrane. Okay, and the endomembrane is this linked series of organelles. Okay, we can see it starting here with the rough ER. Okay, going into the Golgi apparatus, going out here to secretory vesicles, and then eventually can possibly make its way out to exocytose things. Okay, so components of the endomembrane. We got the rough ER, which we were just talking about. This is covered in ribosomes. That's what gives it that rough appearance. Okay, this is the closest one to the nucleus. You can see that nuclear envelope is right there. The rough ER is right there attached to it. Okay, so again, those ribosomes are producing proteins for us. The proteins are then packaged into these little vesicles, similar to the vesicles we saw when we did endocytosis. Okay, and then those are going to be carried to the next step, which will be in the Golgi apparatus. Okay, so those vesicles form by the rough ER. Vesicles carry those proteins to the next step in the process, which is in the Golgi apparatus. And then eventually, those vesicles can empty their contents into that Golgi via a type of exocytosis, not really exocytosis because it's not leaving the cell, but kind of dumping those contents into there, where then those proteins can be modified, kind of put final touches on. And then they're going to go into another membrane bound kind of vesicle, and then they can go some more from there. We'll get to more of that later. Okay, so that was a rough ER. Smooth ER looks somewhat similar, except it doesn't have all those ribosomes bound to it. And it's really a series of tubes with all kinds of different enzymes. And this is where many different parts of the cell are made. Okay, this is what helps our cells make lipids, especially cholesterol, produces steroid hormones for us. So glands like our adrenal gland that produce many of our steroid hormones have lots and lots of smooth ER because those cells are producing those steroid hormones. Okay, lots of smooth ER in the intestinal cells because they have functions in absorbing and transporting fats. Very important for detoxification. So our liver is a big, one of our largest organs as far as detoxifying chemicals goes. And so our liver has a very well-developed smooth ER. Okay, and then also the liver cells, the smooth ER is involved in turning that stored form of carbohydrate that we talked about last time, glycogen, into our blood glucose, and that can be put out into our blood to be used for metabolism. Okay, so that's our smooth ER. The next step in the endomembrane would be the Golgi apparatus. Okay, and I didn't mean to say that the smooth ER is in the middle of those steps at all, but it would go from the rough ER to the Golgi apparatus. Okay, so kind of its primary function is to kind of put finishing touches on proteins that are made by the cell. Okay, so we get this long string of amino acids when we do that translation step in rough ER, and then it's going to immediately fold and kind of get all those levels of structure that we talked about last time. But then it's going to go to the rough ER and get some other things done to it. And some of the things that can be done to it, we can add or trim sugars, we can add or trim amino acids, maybe add a phosphate group, different things like that to make that protein exactly the way it needs to be. Okay, and then at this point, we kind of think of the Golgi apparatus as the mail room of the cell. Okay, mail comes in there, we decide what address it needs to go to, and then it sends it out. Okay, it's kind of the same idea with Golgi apparatus. Those newly made proteins are there, they get touched up, and then they get sent to typically one of three different places in the cell. One could be we need to send that thing out of the cell right now. So that could be a hormone that a pancreatic beta cell is making, like insulin. Right, and insulin's going to get sent out into the blood, be used as a hormone. Okay. It could also just be something that the cell right there needs, right? A protein that's going to help give the cell structure, an enzyme the cell needs for a chemical reaction, many different options there. Or maybe it's a protein hormone like insulin, but the body's not calling for more insulin to be released right now. So we can store up a bunch of vesicles full of insulin. So those are ready to go next time insulin is called upon. Okay. Questions on any of that so far? Okay. All right. So then we got two other ones here that look quite similar. They're really both just these little individual vesicles. Okay. But they contain different types of things. Okay. So lysosomes are full of degrading enzymes. Okay. We kind of think of these as the cell demolition crew. Okay. Enzymes that can break down different parts of the cell, different organelles if needed. Even can destroy the whole cell when it's time for that cell to be recycled. Okay, these bud off of the Golgi apparatus, so degrade cell if needed, and degrade or old organelles or old other components of the cell. So just full of very strong enzymes and chemicals that we wouldn't want just floating around the cell by themselves. Right? We wouldn't want those strong chemicals being able to interact with anything, so we keep them bound up in these little lysosomes. Okay, and then peroxisomes are somewhat similar. They're a lot smaller than lysosomes, but they're also full of powerful chemicals and enzymes. But they're kind of more specifically for detoxifying chemicals, especially free radicals. Okay, so free radicals have ways of oxidizing things, especially oxidizing our DNA, which can cause mutations in our DNA. And so peroxisomes are full of antioxidant type enzymes, oxidase enzymes that can help us deal with those. So protecting cells from those free radicals. Okay. All right, so here's what I want you guys to do for 5 minutes or so. Walk through the different organelles we just talked about, fill in this table, do this with the people on your team. Okay, I'll be walking around if you got any questions. And then we'll come back together in about 5 minutes and wrap it up.
Like shaving off. Why?
Oh, from just the hole punching maybe or something, yeah.
Like sheds everything.
Yeah, that's funny.
And that yes, translate mRNA to beta. I just drew. I drew what the roughly on my thing.
Oh, we don't have mitochondria.
It's on the roughy yard, right? Or, yeah,
This is the rough yard, right?
Yes, yeah. So that's the ribosome slide, but it, you know, that's where you find the ribosomes too. So,
And when we say like they look like anything, it's just.
yeah, that's just kind of more for you to
So, like,
kind of
fish
think about it.
food with-
Yeah, whatever helps you remember what it looks like. Yeah, for sure.
Yeah, no,
Yeah.
I had a bit of fish. I know exactly what you're talking about.
That's funny. All right.
Okay, now smooth it. No ribosomes, flat.
This is the demo crew. Come in and get it done.
It's the mail. Okay. Exercise. You want to shoot that. You're gonna remember it on the exam because of me. And you know it. Yeah, you're gonna be like, you're gonna remember. Lysosomes, they hold the toxic
Yeah.
It has the same thing, except it doesn't butt off of the.
Okay, guys, let's come back together for a sec here. May or may not be completely done with that yet, but yeah, any specific questions that you wanted to ask based on those things that came up as you were checking it out? Yeah.
How often are like organelles regenerated or remade?
Yeah, I think it totally depends on which organelle you're talking about. Yeah, so I don't have a good answer for you on each of them. Certain organelles, like for example, like the mitochondria, are constantly changing, constantly moving. We used to think of mitochondria as like these individual little beans throughout the cell, which is kind of how most textbooks show them. But really what it is kind of this big like glob of kind of like a gel type mass. And so the membranes of it are constantly moving, constantly changing. And so those are being recycled pretty much all the time. Lysosomes are being changed out all the time. The nucleus, not very often at all, right? Probably not much change over at all, except maybe around times of cell division when we're going to change the nuclear membrane. So yeah, I don't have exact numbers for you, but those are just a couple ideas around that question. Yeah, it's a good question. Yeah. Other questions on that? Okay, let me just say a couple things before you start packing up. One thing that I just want you to note, some of the stuff that we do in class is the reason that I have a class set up this way with a bunch of clicker questions, a bunch of things to fill in, is just so you have some time to process things, right? Because I could just drill you with all the information and we could just be done in 20 minutes. But you could also just Google everything if it was all just about information, but trying to set up things in such a way so you have time to think about things, process things, talk about it with the people on your team. A lot of times you have one idea about something, somebody else on your team has a different way to look at it. That helps you understand it, okay? Secondly, with the clickers and with the fill-in things, okay? Don't start packing up quite yet. With the clickers and the fill-in things, there's a lot of educational research around the idea that if somebody makes a decision on something while they're learning, then they'll remember it way better than if they didn't have to make that decision, okay? So the clickers are really important for that because you had to make a decision. Doesn't matter if it's right or wrong. You made a decision, then you found out whether it was right or wrong. That's important. Secondly, when I have things for you to fill in, sometimes there are things that are going to show up later. And so the tendency is to just wait until I show you it and then you write it all in. But that kind of defeats the purpose. Okay, so I would encourage you when more so a little bit later on, we'll have diagrams you're gonna fill in. I would encourage you to, as I'm having you do that with your team, write those things in, make those decisions, cross it out if you need to later once you see the right answers. But making that decision, engaging in the process is just such a better way to learn than just waiting until somebody gives you the answer and then you put it in, because you don't really need your brain for that, right? There's really no learning involved in that, just transferring of the text from the slide to your hand. So just a couple things behind all of that. So have a wonderful weekend, you guys. We'll see you on Monday.
I'm gonna pass all of that.